Hello everyone, I am Francho Nerin and I'm going to tell you about the progress on my project for predicting allosteric sites with machine learning. You have heard already and will hear during this conference how allosteric drug discovery has become of great importance, and also the combination of allostery and machine learning has gained a lot of interest progressively during this past year, starting from the first application of a neural network to model allosteric receptors by Jean-Pierre Changeux here in the audience in 1987. However, computational tools for allosteric drug discovery have some limitations. There are multiple tools that are able to detect ligand binding sites in protein structures. However, allosteric sites must engage in some kind of functional regulation, and for that there are some computational approaches that prove that with models. For example, perturbation methods based on elastic network models that measure perturbations in the dynamics, and also representing the protein structure as networks of residues linked by edges that can contain different information. And also molecular dynamics is a very powerful tool to obtain information about different conformations of proteins. One level of complexity above all this is the use of machine learning to predict allosteric sites. There exist a lot of models now in literature that mainly use a dataset of allosteric sites annotated in 127 protein structures, and that use as features for machine learning information from all three levels of protein information: sequence, structure, and dynamics. And lately also some models incorporate protein language models. And in general, this results in models that use only a selection of all of the potential features that could be used for predicting allosteric sites, and models with varying accuracy and availability. Therefore, the aim of our work is to generate a novel machine learning model for predicting allosteric sites, starting from enhancing the available data and using as much updated data as possible, attempting to use all of the feature types and analyze their combinations, including protein language models, and in the end obtain a public model in which we can measure which features are important for predicting allosteric sites. We can make a systematic benchmark with other existing models in the literature, and also, to our knowledge, one thing that has not been done is to systematically use APO structures to measure the performance of these models in these conformations. This is the workflow that I follow to achieve that, starting from data collection, measuring features in the proteins we use as training data, and then deploying the machine learning models. For this, I use two parallel approaches: one to predict single residues of proteins as to whether they are part of the allosteric site or not, and in a second approach use pockets predicted by a pocket identification algorithm and classify them as to whether they are the allosteric pocket or not. For this task, the main method is machine learning, which aims to learn how to predict a class of the data based on some features. And specifically for this project, I use automated machine learning, AutoGluon, which allows to quickly train multiple models with optimal hyperparameters to select the best. And in particular for this data, allosteric site residues in the protein or allosteric pockets are just a very small number with respect to the number of residues or pockets that are non-allosteric, so we have a high imbalance in the data, and we use these two metrics, MCC and F1 score, to appropriately account for that during training. For features to train machine learning, I have introduced before protein language models. Protein language models are very big neural networks trained on millions of protein sequences that in general they learn the language of proteins, and we use them for our project to obtain thousands of abstract features that we will use to train the machine learning models. In addition to protein language models, we measure features based on single amino acids for each single residue, and also our pocket detection algorithm for the pocket-based models. This provides some physicochemical pocket descriptors. For the residue level features, they cover the 3 levels of protein information, sequence, structure, and dynamics. Now, going into the results of the workflow and starting from the data, I compile a database on more than 3,000 PDV structures that have annotated allosteric modulators and allosteric sites. As data sources for this, I use the allosteric database, which annotates allosteric sites in diverse proteins. Also a data set of allosteric sites in kinases, and also 2 data sets of allosteric ligands developed here in our consortium for ion channels, for the glycine ion channel and for the nicotinic acetylcholine receptor ion channel, and also for the allosterome of GPCRs. Some insights into this database is that most allosteric sites are found in proteins, but there are also some annotated sites on other types of structures, and many types of molecules can be allosteric modulators. However, for our training data, we select only allosteric sites found in protein structures and that are formed by small molecules. And the way we process this data for machine learning is first clustering it to obtain a set of cluster representatives that are all different amongst each other. And then, since we want to compare both pocket and single base models, we keep only proteins that for which the pocket identification algorithm can find a positive pocket. And in the end, we are left with 289 proteins, which might look like a very dramatic drop from the initial data. But we have to take into account that this is still more than double the structures than ASBench, which is the dataset that exists in machine learning models for allosteric site prediction use. In addition to this, we have collected new PDBs on an extra test set, which are all different proteins from the thousands of proteins that we have here, and that will be used for further validation and also to compare with other published models. So in total, I trained 7 models based on residues or on pockets that use different combinations of features, and since they are trained on the same data, they are all comparable amongst each other. For models based on single residues, I train models based on physicochemical features, based on embeddings or on the combination, and also I use a technique called fine-tuning of protein language models. And then for allosteric pockets, it's the same thing except for the fine-tuning, and it also uses the pocket features. So for the results of this, as we can see here comparing both types of models, models based on pockets outperform considerably models based on single site residues, so I'm going to continue discussing this. And among the models that use pockets, the best performing model is model number 5, which uses only physicochemical features. It is interesting to note that when they are combined with protein language model embeddings, they do not seem to be able to help improve the predictions. And so this model 5 is the one that I'm going to keep discussing in my last slides. Some more numbers about this model 5. For our test set of 60 PDBs, we predict correctly 42 allosteric sites. And then for the extra test set of 9 PDBs, we predict correctly 7 of them. And this extra test set is also the one that we have used to compare with other 9 published models. And the results are that our model is the one that performs best, predicting 7 sites, followed by AlloSite Pro predicting 6, Passer, Allo, etc. And the way these predictions look on structures is as follows. So for example, for this luciferase, this is the only protein for which our model is the only one that is able to correctly predict the allosteric site. Then there is also this CFTR target for which our model also predicts a pocket. However, I would like to highlight here, for example, how for this target Passer, the allosteric site that it proposes are the binding sites of ATP, And in the case of the luciferase, the predicted site here is the active site, which is behind the datasheet. And in this other target, the site that our model predicts, it is a phospholipid binding site. And these findings led us to think that maybe our models are biased toward predicting ligand binding sites due to the use of pocket algorithms and their focus on ligandability and druggability. And in fact, when we take a look at the feature importance of what our model has learned, we can see that the top features are F-pocket features based on pockets that measure properties of pockets. However, in a different note, it is also worth noting how among the top features there is also at least one dynamic-based features that may point how dynamics and the dynamics of a site are important for Allostery. However, still the high importance of F-pocket features and the findings on the predicted sites in the proteins I have shown motivated us to look for hypoconformations of the proteins on our test sets and measure the performance of our model on hypoconformations that do not have a ligand that is stabilized in the pocket. And although our model struggles more on the extra test set of 9 proteins, our model still performs the best among the other 9 models, predicting correctly 5 pockets out of the 9. So as a summary, we have doubled the available training data for allosteric site prediction model development from 127 to 289 protein structures. And we have developed a model to predict allosteric pockets accurately. This model performs better than other 9 existing models on our extra test set of 9 proteins. And it also performs better than these extra 9 models on the extra test set, predicting 5 out of 9 pockets. And as a finding on the features to be used for predicting allosteric sites, protein language model embeddings did not seem to be able to predict or improve allosteric site prediction. Thank you for your attention. I will be happy to answer all of your questions.